Hello, I'd like to welcome everyone to the EBTC Scientific Symposium, the first one this year, 2021. So Happy New Year, and I hope you enjoy the seminar. It's my pleasure to introduce the US EPA IRIS team, who have just published their systematic review of effect of phthalate exposures on human health. It's Dr. Erin Yos, Dr. Lena Dischow, Dr. Beth Radke, and Todd Blessinger. So a little bit about EBTC. So EBTC is putting workshops and seminars like this for the last 10 years. We are an international collaboration of various stakeholders who are united in their vision that the environmental health assessment should be done using evidence-based methodology. The statement of our mission and funding is displayed in front of you for transparency. We are governed by the Board of Trustees and have a staff that's managing the working groups that are performing the projects. We have collaborative projects going on with various stakeholders in this field who are listed here. We have a particularly strong relationship with US EPA and European Food Safety Authority, which we're very proud of, as well as NIHS and OECD. These are some of the EBTC projects. They're basically falling in two buckets. One is we do systematic reviews on topics of interest, and the second one is methodological projects, such as critical appraisal tools for in vitro studies and such. As I mentioned, we frequently put together workshops and seminars to bring all the stakeholders together, sometimes from different areas, from different fields, to put together and get consensus on different methodologies relevant to environmental health. We had 2 of such workshops in 2019, and we do hope that this pandemic will end at some point and that we'll be able to meet in person again. And with that, I'll leave it to Erin to start the presentation. Enjoy the webinar.
Thank you guys so much for the invitation to talk today. We wanted to give some of the highlights of our special issue on the systematic review of phthalates, which is something we've published over the last 2 years in Environment International. And it's an offshoot of what was going to be the EPA IRIS assessments of phthalates. So I'm Erin Yost. I'm going to be the first speaker. I'll give the introduction to the Irs program and some of our lessons learned. And then I'll pass it off to the other speakers. We have Beth Radke, who led the epidemiology efforts for the special issue, Laura Deshaw, who was involved with the animal study evaluation efforts, and then Todd Blessinger, who has a dose response model that he's going to present. And I led the animal toxicology efforts as part of the special issue. Let's see if I can advance.
It did not advance.
Did not, I know.
There you go.
Okay, so first of all, some background on phthalates. So phthalates are a class of chemicals that are used in a lot of consumer products like building materials and plastics, cosmetics and personal care products. So it's a lot of things we use every day. And because of that, human exposure to phthalates is ubiquitous across all life stages. And there's been a lot of concern about the health effects of phthalate exposure, and that includes concerns about endocrine disruption. So one of the key pieces of evidence for that in animal models is that when you expose male rats to certain phthalates during late gestation, they get this very distinct phenotype of malformations in the reproductive tract. And that's thought to be caused by phthalate-induced inhibition of testosterone production, as well as inhibition of a few other things. Insulin-like 3 hormone, which is responsible for the testicular descent, and direct targeting of the Sertoli cell. So it's got this mixed mode of action that causes very distinctive reproductive effect in male animals. So there's that concern with endocrine disruption, as well as concern about other health effects that we'll touch on in this presentation. So some background on the Irs program. So we were created in the mid-'80s by the EPA to help foster consistency in the evaluation of toxicity in chemicals across the agency. So what we do is we focus on environmental chemicals, and we do hazard identification and dose response analysis. So our assessments then go on to be used by risk assessors in the field to help inform risk management decisions. And they are also used by EPA program offices like the Office of Water or the Office of Air to help inform their regulations. So we don't make regulations in IRIS, but our assessments do go on to inform regulations. So some history about phthalates in the Irs program. So there are some existing Irs assessments of phthalates that were published in the late 80s and 1990 for several phthalates, but you can imagine that the science has progressed quite a bit since then, and those assessments are pretty out of date. So we were in the process of updating them in 2017, but they were discontinued by the EPA, which is something that happens from time to time due to shifting priorities. And also in 2017, IRIS had just really begun to embrace systematic review methodologies. We had Chris there, who had just joined as our leader, and the phthalates assessments were going to be one of the first published examples of that. So when they were discontinued, we contacted Environment International to see if they would be interested in publishing what we had so we could get our data out there and also kind of showcase our new systematic review methodologies. So thankfully they liked that idea, and we were able to make the special issue. So here's a list of all the studies in the special issue. So they range from, we had 2 papers on study evaluation methods for animal and for epidemiology studies. And then we had 6 systematic reviews of health effects. So 4 of those were epidemiology, and then 2 of them were animal toxicology. And then finally, 1 paper on dose response modeling. So what we're gonna do today, we'll show you some of the findings from these papers. But first, we're going to walk through just a little bit about the what is the IR systematic review method, and what are some of the lessons learned that we came across while making the special issue. So first, overview of Irs methods. So we begin all of our assessments with a database search. So we search databases like PubMed and Web of Science. We search the gray literature, and we'll consult existing assessments for the chemical if they exist. And that will usually be a pretty broad search where we end up with a lot of studies. We then go on to screening. So that's a 2-phase title abstract followed by full text screening. And that's guided by a PICO. So that's going to be the populations, exposures, comparators, and outcomes that we're focusing on for the assessment. So that's going to delineate what animal and human evidence we want to focus on for the assessment. So from that screening, we end up with 2 pools of studies. We have our health outcome studies and PBPK models. So that's the health outcome studies in animals and humans. That's going to be the main focus of the assessment. And then supplemental studies like mechanistic data and toxicokinetics that'll be a supplement to that primary health effect data. So when we have our studies that meet PICO, we then put them through study evaluation. So for that, we use a domain-based approach. And here we have our animal domains and the epidemiology domains. You can see that they're different, but they're both designed to get at the question of looking at risk of bias and sensitivity. So it allows us to articulate the strengths and weaknesses of the study design for each of the individual studies. And we usually do that not at the level of the study, but at the level of the outcome, because there can be different concerns for different outcomes. And Laura DeShaw is going to talk more about our animal study evaluation method. But essentially, we look at each domain, we assign a domain judgment of good, adequate, deficient, or critically deficient. And then we look across all those domains to come up with an overall confidence rating for the study or for the endpoint. So that's going to be high, medium, or low confidence or uninformative. So once we have our study evaluations, we then go on to evidence synthesis. So we do that using adaptations of the Hill considerations. This slide just gives a rundown of what those are. So we look at consistency, and if there is inconsistency, we think about what could be causing that. So we have to think about, we think about it in terms of what were the confidence in the studies that we determined during the study evaluation, the sensitivity, things like the populations or species that could affect what kind of health effects you're observing. The magnitude and precision of effects. So if you have a large magnitude and very small variation, that can increase your confidence in the results. And that could be individual studies or a meta-analysis. We look at a biological gradient or dose response. So if you see that pattern of effect, it can strengthen your confidence in the results. We usually think monotonic effects, but it can sometimes be a non-monotonic effect depending on the endpoint. Coherence, so that's looking at related endpoints within and across studies. So for example, like if you saw effects on testosterone and effects on reproductive organ weights, then that would be coherence because those two effects are related to each other. And then for epidemiology studies, you could also might have natural experiments or examples of temporality, like a temporal relationship between exposure and effect that would strengthen your confidence. So we do this separately for animal and for human evidence. And we may also do a synthesis of mechanistic evidence to support our animal and human syntheses. So using those Hill considerations, we then develop separate judgments for the animal and for the human evidence. And we assign an overall descriptor. And this slide shows what those descriptors are based on the strength of evidence. So our strongest evidence, we would rate that as robust. And we have here our definition of what we would call robust. So that's going to be a set of high or medium confidence studies that show a consistent pattern of effect, and they're strengthened by some of those Hill considerations. So as the evidence gets more uncertain or more limited, we would reduce that rating to moderate or slight, and then indeterminate would be an instance where we don't think we can come to a conclusion. And then finally, compelling evidence of no effect would be a pretty rare instance where there's a large database showing that a chemical does not cause a particular health effect. So once we have those separate conclusions for the animal and human evidence, we then integrate across those to reach an overall judgment about the potential for a chemical to cause a particular health effect. So we would do that by considering things like the human relevance of the animal and mechanistic data and coherence across those two lines of evidence. And then we assign an overall a judgment, which we show here on the slide. So that's not something that we did for the special issue on phthalates, because we just weren't there yet with the assessments when we when the assessment was abandoned and we started writing the special issue. But it is something that we do for IRIS assessments. And the strongest evidence judgments are the ones that would then go on to have a toxicity value derived. So now for some lessons learned. So as I said, this was really one of the first systematic review efforts by IRIS. So we learned a lot along the way, and we're implementing new practices in our current IRIS assessments based on some of these early experiences that we had. So the first lesson relates to how to prioritize outcomes for systematic review. So chemicals that have large databases like phthalates, like a lot of chemicals that we work with in IRIS, often report data on a lot of different health outcomes. And some of them have a lot of data, some of them have a little data. And it's really time consuming to do a full systematic review on every single health outcome that's been associated with a chemical. So it's pragmatic to focus on outcomes that we think are going to be most important for risk assessment. So what we did for phthalates was we took a pretty informal approach where for the epidemiology studies, we included all outcomes in the PICO, but we did full systematic reviews only on a subset that had sufficient data available. And then the animal studies, we focused just on 6 health outcomes that we thought would be the most important for the assessment. So male and female reproductive, developmental, liver, kidney, and cancer. And I will say the reviewer comments on the animal papers, pretty much everyone commented on this limited PICO. It seemed like cherry picking data. They thought we were potentially leaving out some important health outcomes for phthalates. So what we're doing in the future and also in our current IRIS assessments is using systematic evidence mapping as a way to prioritize outcomes for full review. So here's an example of what an evidence map looked like. So the evidence maps are essentially just summarizing what kind of data you have. They could include study design information or maybe some initial summary of effects, but they don't go so far as to draw a conclusion. So this is an evidence map for animal studies of vanadium, which is one of our newer IRIS assessments that we're developing right now. So you can see the heat map at the top. We have studies binned according to the duration of exposure, what species were evaluated, and then what kind of health effects they were looking at. So you can look at this and you can see where your evidence lies. You can maybe eliminate some health outcomes. You can see that they just don't have the information to support any kind of conclusions. And you can make it public with your assessment material, so you maintain that transparency about why you decided to focus on certain things. And I really like the evidence maps. It takes a lot of upfront work to create them, but it really helps with planning for the assessment, for on our end, assigning staff and finding the expertise you need to finish the assessment. And then as well, you get that transparency with the public. So this is animal data, and this is an example of an evidence map of epidemiology data for PFAS in this case, which is, we've got some of the effect estimates extracted from some different studies. So our second lesson relates to incorporation of mechanistic evidence. So what do we talk about when we're talking about mechanistic evidence? So that's going to be data from experimental studies that is usually endpoints occurring at the molecular or chemical level that are not considered to be adverse on their own. Maybe they're precursors to adverse effects, but they're going to be informative of your health effects evaluation. So that can be data from in vivo or in vitro models, as well as in silico models, maybe some non-mammalian species that are used as alternative models. So mechanistic evidence it's very diverse. We usually have a lot of it. It usually vastly outnumbers the animal and human studies that meet our PICO criteria. So it can be very overwhelming to deal with in a systematic review. So what we did for phthalates was we, again, took kind of an informal approach. And when there was mechanistic data that was mentioned in one of the in vivo studies that met PICO criteria, we kind of wove that into our synthesis as a way to augment our conclusions. So for example, if you look at diisobutylphthalate, our synthesis of effects on testosterone, there are some studies that looked at effects on genes in the steroidogenesis pathway. So we just, put a couple sentences in our synthesis about that to help support our conclusions. So I'd call it a pretty informal approach, not very systematic. So what we'll do in the future is that we're trying to pivot towards a more transparent and operationalized method of identifying and including mechanistic data in our assessments, while maintaining a lot of flexibility and kind of a fit for purpose methodology. So what we find is that not every not every chemical, not every health outcome needs a full analysis of mechanistic data. For example, if you already really have strong animal or human evidence, then mechanistic data is not really going to change your conclusions. But if you have some uncertainties, then maybe mechanistic data can help fill those data gaps. So we have to maintain that flexibility, but be very transparent about it. So one thing we're doing is that we're making inventories of mechanistic data, and that's kind of another evidence mapping exercise that we would do at the outset of the assessment. So here is a literature tree for one NatFol, and you can see it's got, this shows the whole database. It's got the included studies, human and animal, the excluded studies that weren't relevant at all, and then the supplemental material. We have different categories. One of those is mechanistic, and within mechanistic, we have it binned according to what type of molecular events it's looking at. So that's just another way for us to get organized up front and to be transparent about what kind of data we have. And we do actually have an example for phthalates of one way to approach a mechanistic analysis and a systematic review. And this was not published as part of the special issue, but it is an offshoot of the same project. And it was written by our colleague Xavier. And it essentially takes an adverse outcome pathway approach to looking at species concordance for the male reproductive data for dibutyl phthalate. So AOP approaches, probably a lot of people here are familiar with it. It just looks at the key events that link a molecular interaction to the ultimate effect of the organism level. So what he did is he took the this AOP or MOA for the phthalate-induced effects leading to loss of male reproductive function. And you can see that there are effects on the Leydig cells and effects on the seminiferous cord and Sertoli cells that that lead to a series of key events that cause those effects. And so we took each of those key events and used that as a way to organize the data from animals and the data from these human testicular tissue models. So you can compare and contrast the effects across species and then compare those to humans. So that it's a really great way to organize your evidence and support your conclusions about human relevance and about species concordance. This is a really in-depth analysis. It's not something that we would do for every IRIS assessment, but it's one way to approach it when you do have that mode of action data available and you have a large mechanistic database that you think can be useful for filling some data gaps. So I'm going to pass it off to Laura Deshaw, and she's going to talk about our study evaluation framework.
Okay, thanks everybody. So I'm just going to give a brief overview of the study evaluation strategy that we have for animal studies. Next slide, please. So Irs gives you a big overview. It was developed to fit the needs specifically of IRIS assessments. The general approach is the same for human and animal studies, although the specific domains that are evaluated are slightly different across each of them. As Erin already mentioned, judgments are made at the outcome level, and we look at strengths and weaknesses related to reporting quality, internal validity, and sensitivity. And we've developed a framework of core and prompting questions that are used to help guide the reviewers to look for the specific study attributes or elements that we want them to be considering for each domain. But within that general framework, they're asked to use subject matter expertise. And that's used to develop chemical exposure and outcome specific considerations as needed for a specific assessment. And all of the rationales are documented and publicly available in HAWK. Because there may be very specific needs for the evidence base at hand, The process undergoes criteria development and pilot testing, where the reviewers will go through a subset of studies before they go through the full study evaluation. Then afterwards, there's a conflict resolution step to allow them, if there are differences across reviewers, to kind of discuss and come to a common understanding and reach the final consensus judgments. Next slide, please. So Aaron already kind of overviewed this. We have the individual domains. For each domain, there's a judgment of good, deficient, or good, adequate, deficient, critically deficient, and those will lead to an overall confidence rating of high, medium, or low confidence, or uninformative in rare cases for animal studies. Next slide, please. So the next series of slides will have this same general format. We'll have the core question at the top, the general considerations on the left, and then an overview of the results from the phthalate studies, animal studies on the right. So for reporting quality, we're asking, does the study really report the information that's necessary to evaluate the study? And we have developed kind of two groups of information. There's critical information, which is essentially the minimum necessary to move forward with the study evaluation. And if any piece of that critical information is missing, we would rate the domain critically deficient, and the study would be considered uninformative unless we were to get more information. And then there's important information, which is really everything else you would want to know and is used to distinguish between a rating of good, adequate, or deficient. So in this case, we had most of the studies were rated good or adequate. The good is the dark green, adequate is the light green, yellow is deficient, and red is critically deficient. There's also a not reported, which I'll get to in a later slide. So in cases where we had adequate, there was some information missing, but it wasn't necessarily considered to be critical. For deficient, we had a couple studies where the sample size was not reported anywhere, and another one where the study report was missing all of the methods information for the endpoints. Next slide, please. Okay. Allocation, where the animal is assigned to experimental groups using a method that minimizes selection bias. This is one of the domains where we will often see, and this is really getting at whether or not randomization methods or normalization methods were used. This is one of the ones where we'll often see not reported. So that's denoted by the 16 on the right. In general, most of the studies were rated adequate, meaning that they randomized. They said that they randomized, but didn't describe kind of the method of the randomization, or they normalized by body weight or some other factor. In one case, in pretty rare case, we got a deficient. In this specific case, the control group included 5 pairs of sisters, which was considered a risk of bias concern since the animals were not fully independent of one another. Next slide, please. For observational bias or blinding, did they implement measures to reduce this? This is another one where it's almost more common than not for the study report not to report information about blinding or other methods of controlling for observational bias. If the study did not report the information, the authors were contacted. Generally, when they responded, they said that they did address observational bias, and the ratings were adjusted to reflect that additional information that we received. For not reported, we asked the reviewers to either interpret it as adequate if it's the type of outcome where there's less concern for observational bias, or interpret it as deficient if there's a higher concern for observational bias. Next slide, please. Oh, sorry, I didn't notice that it changed. Okay, confounding. So are variables with the potential to confound or modify results controlled for and consistent across all experimental groups? In general, the phthalate studies were pretty well conducted. We didn't see any, there weren't any noticeable inconsistencies based on the study report or uncontrolled variables. So these were generally rated good or in a few cases adequate. Next slide, please. Did the study report results for all pre-specified outcomes and tested animals? Here we're looking for just any kind of outcome. It could be any type of results, either qualitative or quantitative. In general, it's pretty good. And there might be some omissions or some attrition that's unexplained, but it's not, it's expected to have significant impact. We did have some deficient ratings for this domain where the sample sizes were not clearly reported in the methods, and there were pretty large variations across the different outcomes or treatment groups. So in this case, it was really difficult to determine whether or not attrition occurred. So these studies, these specific studies were rated deficient. Next slide. So the next domain is really asking whether or not the study adequately characterized the exposure to the chemical of interest and the exposure administration methods. And this is something where in the early phases of the pilot testing, it's really important to nail down what the specific needs are for your chemical or your evidence base. And you'll want to make sure that the reviewers are on the same page. So things that you might want to consider are the purity, the stability of the chemical. If it's known to have impurities that could be drivers, that's something that you would want to know. Or if you have a chemical that's unlikely to be stable in solution for very long, you would want to know, have the reviewers note whether or not they're making it fresh reasonably enough so that it's stable throughout the exposure. Some other additional considerations that were used for the phthalates are for gestational exposures. Did they account for dam weight gain? For dietary exposures, did they measure food consumption? Because there could be some palatability issues where in the higher doses where they might not actually be getting the nominal dose. There were also some low dose studies that didn't verify the dosing concentrations, and these were rated deficient. Another specific thing with phthalates is they're very commonly used chemicals, and so there can be relatively high background levels, which is a potential concern with really low dose studies. Next slide, please. With exposure, timing, frequency, and duration, was it sensitive for the specific outcomes of interest? This is another one that's very highly outcome dependent. And an example of this with the male reproductive outcomes, Erin already alluded to it earlier, is there's this masculinization programming window, which occurs from gestational day 14 to 18 in rodents. And if this window isn't covered by the exposure, you're probably not going to see any effects of the exposure. So it was really important for male reproductive outcomes to be to make sure that this critical window of sensitivity was covered. But in general, for the phthalate studies, they were pretty well conducted. Next slide. Sensitivity and specificity were the procedures sensitive and specific for evaluating the outcomes of interest. Another very highly outcome dependent domain. Things to consider include what was the timing of the outcome evaluation relative to the exposure? Was there sufficient time where you would expect to see an effect on that particular outcome? And the validity of the assay for evaluating the outcome of interest. So if you have an assay that's not sensitive for that particular outcome, that might be a consideration. So a very specific thing with testis histopathology that we've learned is that the fixative is really important. So formalin is a really common fixative. However, with testis tissue, it's known to introduce artifacts that can reduce the sensitivity. So in the case of a study where formalin was used to fix testis tissue, that would be likely rated deficient. Next slide, please. So the last domain we have is results presentation. Are the results presented in a way that make the data usable and transparent? There are a lot of different things you want to think about. Are there limitations in the presentation of the data? Are they providing quantitative results, or are they only having providing a qualitative description of the results? Is there inappropriate pooling of the data across groups or across sexes, et cetera? So in general, studies where, ideally we would have quantitative results for all of the outcomes of interest, but that's not how life works sometimes. So in cases where there was a qualitative description, but there was no treatment-related effect, those were rated adequate. For deficient ratings, there were cases where the less preferred metric was used. So for testis weights, absolute testis weight is the preferred metric, but if the study only presented relative to body weight, that was considered deficient. For reproductive or developmental effects, only presenting individual animal data is a limitation because it doesn't allow you to account for litter effects. In one study, there was a critically deficient rating, and this is where there was directly conflicting results between, I think it was a table and a graphical representation for the testosterone measurements. And so that was rated critically deficient for that specific outcome. Next slide.
So Laura, sorry to interrupt, just to let you know, you have about 2 minutes left on your presentation, and then
I'm wrapping up. All right, so we have our overall confidence judgment. Are the results present? Oh, sorry, I forgot to change that. So in general, most of our studies were evaluated, were given a higher medium confidence rating overall. Low confidence ratings were generally occurred when there were multiple domains that were rated deficient, although in some cases it could be driven by a single deficient domain rating. For example, the testis histopathology, if they use the incorrect fixative, that might be enough to drive a study to be considered that outcome to be considered low confidence overall. Uninformative ratings were very rare and driven by that one study where there had the critically deficient rating and results presentation. Next slide. So last slide, lessons learned. There are some advantages, there are some challenges. The main advantages were that the outcome level evaluations reduce the potential to over or underestimate the confidence in the results versus a single study level judgment. It addresses sensitivity in the study evaluation as opposed to trying to address that later, perhaps in the synthesis. And there's flexibility because it uses subject matter expertise within the general framework. And so it can be more easily adapted to the needs of assessment in a particular assessment versus a set of rigid criteria. Challenges are, however, that the process is really resource intensive because you need at least 2 subject matter experts per study, and it can take a really long time depending on the complexity of the study. And there are potentials for inconsistencies in the judgments across reviewers or across assessments, but that is what the pilot testing and conflict resolution stages. But again, those add more to the time investment. It is an evolving approach. We're in the process of trying to improve and refine it. We're not saying that it's perfect, but it's what we have now. And we're hoping to build upon previous work to streamline future assessments. The evaluation tool that we're using, Hawk, has gone through some updates that improve our communication and our transparency since the phthalates were done. And then another big takeaway, I think, is that, at least for animal studies, critically deficient or uninformative ratings are really very rare. It's not like we're throwing out studies left and right, which I think there were some concerns about. That's all I have. Thanks.
Very good, thank you, Laura. Erin, I think you're still driving, so the next presenter, if I understand correctly, is yourself, I think. Erin, I think you might be on mute.
Hi, sorry about that. All right, so I'm gonna give you a rundown of some of the highlights of our animal toxicology systematic reviews. Sorry, the slide advancing has been all kind of funky today. So, okay, so we did systematic reviews on 2 different phthalates, and these 2 are just the ones that happened to be closest to finished as IRIS assessments when the assessments were terminated. So that's why we have these 2 particular ones as our papers. So diisobutyl phthalate is used as a substitute for dibutyl phthalate, which is being increasingly banned because it's known to be a pretty potent developmental toxicant. But unfortunately, DIBP has actually been found to share that same anti-androgenic mode of action as the phthalates that it's replacing. And then diethyl phthalate, which is the most widely used phthalate in cosmetics and personal care products. It's a low potency as a developmental toxicant, and it generally has been shown to not have that same anti-androgenic mode of action. So just really quick on our literature search, we searched a few databases, we did some great literature searches, title abstract followed by full text screening. So our PICO, as we were looking for mammalian animal studies that were exposed to these phthalates or their primary metabolites by the oral, dermal, or inhalation routes. And they had to have a vehicle only or untreated control. And again, we were focusing on these 6 outcomes that we thought were going to be most important. And so here's what our literature flow diagram looks like. So here's the example for DEP. So it shows going from all the studies that we found in the database searches down through the title abstract screening, followed by full text screening, and then ultimately the 39 studies in this case that we found that met PICO criteria. So some highlights from those papers, these are directly out of the papers. So for diisobutyl phthalate, we found that our conclusions did agree with the fact that it's it seems to be a male reproductive toxicant. It also had some evidence of female reproductive toxicity and hepatic toxicity, but those are less studied, and there's a little bit more uncertainty about those. For DEP, we found, as is usually thought about this chemical, that it was not a very potent developmental toxicant. It didn't affect testosterone in fetuses, but it did have some effects on sperm, which were unexpected. And we had some interesting observations about the low dose studies. So I'm going to focus on DEP for the rest of my slides here. So this is the heat map summarizing the substudy evaluation results for DEP. So we have our domains going down this axis, and then the ratings for each of them. And then the bottom row here is the overall confidence rating of high, medium, low, or uninformative. So you can see that probably about half the studies were well conducted. We had no major concerns, and we gave them a high or medium confidence rating. We had one that we found to be uninformative because the results were almost impossible to interpret. So we eliminated it based on that. And then there's you can see a large swath of studies that Laura mentioned that are these low dose studies that didn't verify the doses that they were working with. They didn't do any analytical verification. And these were all conducted by 2 different laboratories. So we rated them deficient in that domain and then low confidence overall. So I'll come back to those in a minute. So just to highlight some of our results for male reproductive effects. So looking first at effects on fetal testosterone, we had 3 studies. 2 were high confidence, 1 was medium confidence. This is data that we extracted ourselves into HAWK, which is a program that we use to make our study evaluations and our data sets publicly available. And it just shows whether or not there was a significant effect at each dose level. So for DEP, 3 studies, they saw no effects. And if you zero in on the study that looked at multiple dose levels, you can see no dose-related response whatsoever on testosterone up to really high dose levels of almost 1,000 milligrams per kilogram per day. So this is our evidence profile table. It's a structured framework that we use for synthesizing our evidence. So we had these 3 studies. The factors that increase strength are consistency, the lack of effect in a sensitive species, as well as minimal concerns for bias and sensitivity in these high and medium confidence studies. So that really supports a lack of effect, but we have so few studies, we can't make a conclusion as strong as saying that DEP doesn't have any effect on testosterone in fetuses. So we ultimately landed on the conclusion of indeterminate. So now we look at postnatal and adult exposure effects on testosterone. We had 5 studies, and actually 3 of them saw decreased testosterone, but 2 of those were low confidence studies. So we had some concerns about the conduct of those. And then the high confidence study that saw decreased testosterone, if you look at the actual data, you can see that there is a really large amount of variation in each of these dose groups in a small sample size. That kind of decreases our confidence in that finding. So our synthesis for testosterone after postnatal exposure, we had that consistency in the direction of effect, but that low precision in the high confidence study, and then concerns for bias and sensitivity in the low confidence studies, as well as some mechanistic evidence that supported the lack of effect in steroidogenesis. So from that, we arrived at a conclusion of slight. So lastly, effects on sperm. So there were 5 studies that looked at this, and actually all of them, 4 out of 5 saw that there were effects on sperm quality or quantity. There was a 5th study that reported no effects, but they had no quantitative data. So that's kind of limited interpretation. And so the actual effects differed across studies. Some were effects on concentration, some were effects on morphology or motility, but this trend of sperm being affected was consistent. So our synthesis for sperm parameters, we had minimal concerns about these studies. They were high and medium confidence. There was some mechanistic evidence showing oxidative stress that's supportive of an effect, but then there was an unexplained inconsistency in which one study showed no effect at all, and then the actual parameters were different across studies. So, but because of just that consistency, we gave it, well, because of that same direction of effect across studies, we arrived at a conclusion of moderate. So this slide just looks across all of the endpoints for male reproductive effects. And so as mentioned, there is slight evidence for testosterone. There was moderate evidence for sperm. Everything else was indeterminate because there just wasn't enough data, or because effects were too, either there were no effects or we just couldn't conclude anything. But because we had that evidence for sperm, we arrived at an overall conclusion of moderate evidence that DBP is a male reproductive toxicant. And this is interesting because if we go back to this MOA slide for the anti-androgenic phthalates, you can see that they operate through this dual mode of action. So there's effects on Leydig cells and effects on the seminiferous cord. And those are thought to be two independent modes of action. They're separate from each other. So we think it's possible that DEP could be operating through that effects on the seminiferous cord and on Sertoli cells and germ cells that is occurring even though it doesn't have that Leydig cell effect of decreasing testosterone. So we can't conclude that from our review. That's just a hypothesis, but we kind of identified that as an area for possible future research that we thought was interesting. So going back to Laura had mentioned that we had some studies that didn't verify their dose levels. They looked at relatively low doses of DEP, and we had some concerns about that since phthalates are used in everything. There were some dose range finding studies that indicated elevated levels of DEP in the control dose groups. So this lack of analytical verification made us doubt that the ranges were actually what they said they were. And the studies also had some other concerns, like presenting litter mates as individual animals, which can inflate the significance of your results, and then just generally poor reporting quality. And we ended up rating them as low confidence for all outcomes. And these studies also happened to find really profound effects that were not observed in studies that tested higher dose levels. So for example, this is effects on postnatal survival. There were 2 studies that we considered high confidence that saw no effects. And then the low confidence, low dose studies saw this decrease in live pups, increase in dead pups at this low dose level. And then if you look at hepatic effects, for liver enzymes, there was a high confidence study, actually 2, that saw no effects. And then these low confidence studies saw consistently elevated liver enzymes. So there is this evidence of low dose toxicity for DEP, but we don't put a lot of stock in it because these studies had these concerns that we identified during study evaluation. So we flagged that as an also potential area of research if we, it would be good to have some well-designed studies that were examining these potential low dose effects. So I will turn it over to Beth Radke, who did the epidemiology analyses.
Thank you. All right, so I'm going to try to get through this as quickly as I can. The process for the epidemiology studies was a lot like what Erin just described for the animal toxicology studies, with just some tweaks for the specific discipline that we were looking at. So for the epidemiology studies on the literature search, rather than doing individual searches for each phthalate, we did one broad search for the 6 phthalates that we were interested in. So the reason for that is that most epi studies of phthalates look at multiple phthalates. You know, they do biomarkers in urine or blood of a whole slew of them, and so it's easier to identify them all together. We had a really broad PICO. You can see really any human population, any exposure to these phthalates, any comparison, any outcome. Next slide. For the systematic reviews, we focused on these 4 health effect categories: male repro, female repro and developmental, and metabolic effects and neurodevelopment. That was primarily based on data availability. There were a couple of other health effects that probably could have sustained a systematic review, like immune effects and thyroid effects, but we had to stop working on our assessments, and so those got shelved. Okay, next slide. So here is the literature flow diagram for one of the systematic reviews, the metabolic effects. Obviously, in the next few minutes, I'm not going to have time to go into detailed information about 4 massive systematic reviews. So I'm just going to try to give you a couple little tastes of what you'll find there. So this is the metabolic effects. You can see that there were 72 total studies that touched on any of those effects out of the 416 that met the PICO for any of the phthalate health effects. Go ahead, next slide. For study evaluation, you've already heard what that looks like for the animal studies, and I'm not going to get into a lot of detail about how we go through each domain for the epi studies, but I did want to touch on our development of outcome-specific criteria. So we have, just like with the animal studies, we have core and prompting questions, and then we also have a criteria that sort of apply to all outcomes, or at least most outcomes and exposures. But then in addition to that, we develop criteria that are specific to the exposure. So we have criteria for developing or for evaluating phthalate exposure. And then we also want to evaluate the outcome. But, we're looking at a wide variety of outcomes in these set of systematic reviews, and there's no way that the epidemiologists involved would be able to bring expertise on all of those. And so we worked with some subject matter experts to develop these outcome specific criteria to let us identify some sort of those key considerations that we needed to be thinking about in the evaluation. This is the paper that describes the process for developing those. Can you click? And then this is an example of, sort of the criteria for what a good study of diabetes or insulin resistance would look like for each domain. Just to highlight a couple of those, if you click once again. So for exposure measurement, for diabetes, we say that exposure must be measured before diabetes onset. That would most frequently be in a prospective cohort, though it's not required. And the reason for that is concern for reverse causality. It's, with diabetes, there's the possibility of metabolic changes that could influence the amount of phthalates in the body, as well as behavioral changes following a diabetes diagnosis that might change your level of exposure to phthalates. Whereas for insulin resistance, we are a bit less concerned about those issues. We can think that it can be measured concurrent with the outcome because you can have sort of short-term changes in the blood glucose and insulin levels. And if you click again. Similarly for participant selection, for both diabetes and insulin resistance, we say that we must exclude individuals that have known diabetes for baseline in the diabetes and just in general for insulin resistance. With diabetes, sort of the same reason as for exposure, and for insulin resistance, it's because you can't interpret the results of the insulin resistance analysis if you have people who are being treated for diabetes in the analysis. Okay, next slide. So here's a heat map of the diabetes studies. You can see that we actually only had one study that did measure exposure prior to onset of diabetes, and 6 studies that measured it after development of the outcome that were excluded. This is a sort of an unusual number of studies to be excluded. And I think in a lot of systematic reviews, they may be set aside at the PICO level, or we might in the future just kind of set them aside at the refined evaluation plan level, where we look at our systematic map and kind of identify issues that we can save ourselves some time in the study evaluation step. Next slide. And then here's the same thing for insulin resistance. You can see we had a lot more studies for insulin resistance, and they were mostly medium and low confidence. One reason, if you look across all of the systematic reviews for the epi evidence that we don't have a lot of high confidence studies, is that most epi studies of phthalates look at one single measure of phthalate exposure in urine, and the repeatability is not great over time. And so we just don't, we don't have a ton of confidence that we're really accurately characterizing exposure in people based on one measure. Okay, next slide. Okay, so here's some of the results for diabetes. You can see that there, this is just that one study, the Sun et al. Paper, and it focused on the Nurses Health study. They had 2 kind of subgroups: the original Nurses Health study, and then Nurses Health 2. For DEHP, they had results from each of those analyses and a pooled analysis, and you can see that all of those are showing they're in the same direction. They're showing increased odds of diabetes with higher DEHP exposure, but the increase is sort of non-monotonic in the Nurses Health Study 2, and as well as they're just there's imprecision in the estimates. They're not statistically significant. Whereas for DBP, there's only in the Nurses Health Study 2 did they look at DBP alone. but you can see that there's a large effect estimate and odds ratio of over 3 that is highly statistically significant. So in both cases, we have some evidence of an association with diabetes, but looking slightly different. Okay, next slide. Here are the results for insulin resistance for both the Ehp and Dbp. I don't expect you to see the numbers, but you can kind of see the shading. So the dark shading is reflective of an association that's either statistically significant, or there's a clear exposure response trend, or a large effect estimate. The light shading is just anything else that is kind of in the same direction. And you can see that for DEHP in adults, which are the kind of the top two groups there, almost all of the studies are showing an association between DEHP exposure and higher levels of glucose, insulin, and this measure of insulin resistance, home IR. Whereas for DBP, it's a little bit more scattered. There is some evidence, but it's less consistent. Next slide. So just to look at both of them and kind of think about the coherence between them. For DEHP, we have pretty consistent evidence for insulin resistance that's supported by some imprecise estimates for diabetes. Whereas on the next slide, you can see for DBP, we really had that one strong positive association with diabetes, but inconsistent evidence with insulin resistance. But in both cases, we consider that to be moderate evidence, because we have strong evidence for diabetes, for DBP, and for insulin resistance and DEHP that sort of are the drivers. For the other phthalates that didn't see an association with SUN and had even less consistent evidence for insulin resistance, we're calling them slight, so for DEP and BBP. Okay, next slide. And then I just wanted to touch briefly on meta-analysis. If you look through the systematic reviews, you'll see that most of them do not include a meta-analysis. And the reason for that is really that for most of the outcomes, the evidence really just wasn't appropriate for a meta-analysis. They're either, drastically different populations, or they looked at slightly different outcomes, or the analyses were different. But in the neurodevelopment paper, you can see that essentially all of the studies that looked at neurodevelopment, cognitive and motor endpoints in small children, so less than 4 years old, use the Bailey scales, and we considered them similar enough to do a meta-analysis. And you can see some of that here. This is the psychomotor scores, where really only BBP was the only, for motor, was the only comparison that was, had a consistent evidence across studies and a significant association on the meta-analysis. Okay, next slide. And then just to sum up all of the systematic reviews, each one has a heat map at the end that shows the level of evidence for each phthalate and outcome combination, as well as for the overall for male repro. And so you can see that the evidence was robust for male repro for DEHP and DBP for the most part. I think those were the only robust, and then the rest was smattering of moderates, mostly slight and indeterminate for a lot of those combinations, and in some cases that's because of lack of adequate data. Okay, and I think that that's it. So I'll hand it over to Todd.
Todd, are you there?
I can't hear Todd.
Yeah, me either.
So I think Erin or Beth, if anyone on your side. Oh, hello, Todd's here. Excellent.
Hi, can you hear me?
You've got
Yes,
a
you
lot
have
of
an
echo.
Can you hear me now?
That's much better, yeah.
Can you hear me?
Yes.
Yes, can you hear us?
Can you hear me?
Yes. Todd, can you hear us? Unfortunately, Zoom seems to be having a lot of audio problems today. Todd, can you hear us?
Can you hear me? Yes, I can hear you. Can you hear me?
Yes, we can hear you now.
Sorry, I'm just going to use my computer. I was on the phone. It wasn't working for some reason. Okay. So I'm going to be talking about the ordinal dose response analysis of the phthalate syndrome. Next slide. Okay, so the objective of this analysis was to incorporate severity level in the dose response modeling of the phthalate syndrome. So the phthalate syndrome is a collection of related male reproductive developmental effects observed in rats after gestational exposure to phthalates. So it can include effects such as nipple retention, undescended testes, oligospermia, et cetera. Next slide. So historically, the phthalate syndrome is defined as a dichotomous endpoint. So basically, it's an animal is designated as having the PS if it has at least one of the PS designated effects. And only one dose response analysis is conducted on that endpoint, rather than conducting a separate analysis for each individual endpoint. Now, the reason that they do it that way is because different animals react to the phthalates differently. So some might have undescended testes and some might have oligospermia, et cetera. And the rates of each of those individual endpoints might be low, like 10 or 15%. But if you look at the percent of animals that have at least one of the reproductive effects, it's likely to be a lot higher. So if you model each endpoint individually, it's going to dilute the effect, and you're going to underestimate the risk. But what we did in our article is we modeled the PS as an ordinal endpoint, that includes 2 levels of severity based on impact on fertility. So next slide. So we modeled data from a study by Salenfay done in 2008 on DWP exposure to rats. And here are the characteristics of this study. The important thing to note here is that we had individual animal data. Salenfay was the co-author on the article. She provided us the individual animal data. And because we had that, we were able to assign figure out which animals had which effects. So if we had only the summary data, we would not have been able to do that. So having individual animal data made the analysis possible. Next slide. So here are some of the endpoints that were included in the phthalate syndrome in our definition. These are just basically the most common ones, like nipple retention, hypospadias, and histopath endpoints like tubular necrosis, and then epididymis, azone oligospermia, which are measures of sperm count. Next slide. So here's how we define the ordinal version of the phthalate syndrome. So the dichotomous version is basically what I said earlier. So it has 2 levels, 0 and 1. So level 0 is an animal had no PS endpoints. Level 1, it has at least 1 PS endpoint. The ordinal definition, I should step back and define what ordinal means. What I mean by that is it has more than 1 level, and the levels have order. So it's scored like a lesion would be. Like lesions are often scored as minimal, like none, minimal, moderate, and severe. So you have multiple categories, and there's an order to them. So so for the ordinal PS, we had 3 levels. So level 0 again is an animal had no PS endpoints. Level 1 is it had 1 or more PS endpoints that are associated with 0 to moderate expected impacts on fertility, but it had no PS endpoints that were associated with severe impacts on fertility. And level 2 was an animal had 1 or more PS endpoints that are associated with severe infertility. So next slide. So the endpoints that were associated with severe and non-severe impact on fertility. So basically, for endpoints that associated with severe infertility were all the developmental reproductive effects besides nipple retention, and then just a few like real severe histopath occurrences like grade 5 oligo and azospermia in both epididymes. So it had to be pretty severe in that case, but all other endpoints or combinations thereof were determined to have 0 to moderate impact on fertility. Next slide. Okay, here is the model that we used. I'm not going to go over this in much detail. I'll just say a few things about it. First, we used an extension of the logistic model to the ordinal case. And we had to have 2 different curves, one for each level. So when you're modeling a dichotomous endpoint, you typically have probably y equals 1 given dose x is some function of x. But for the ordinal case, we had to have one for levels 1 and 2. So we had to have 2 curves. And we modeled the probability that y greater than or equal to j for j equal 1 or 2. So probably y greater than or equal to 1, which is the same thing as y equal 1 or 2, and probably y equal 2 as a function of dose. And the probability that y is equal to 1 or 2 is the same thing as an animal having at least 1 PS effect. So basically having the dichotomous version of phthalate syndrome. And the nice thing about doing this way is, so basically you see that in the formula, theta is the log of the benchmark dose. So that's the dose that we're aiming to estimate. And we can do this at different benchmark response levels, BMRs, for the different levels. And that's what we did. So you go to the next slide. So we used a benchmark response of 5% extra risk for level 1 and 1% extra risk for level 2. So we had a lower BMR for level 2 because it's more severe. Next slide. Okay, so here are the incidence rates for across the doses for the different levels, level 0, 1, and 2. And also in the rightmost column, you have the incidence rates for levels, an animal having level 1 or 2. So as expected, for the control animals, none of the animals had any PS effects. And then as dose increased, more and more animals had PS effects. And for the highest dose, they all had at least 1 PS. All the animals had at least 1 PS effect, and most of them had a level 2. Next slide. Okay, so here's a plot of a scatter plot of the response rates for each dose. You see those are the dot. If you can read that, the dots in those plot. There's the plot on the left is the probability that y is has level 1. An animal has level 1 or 2, and the plot on the right is the probability of an animal having level 2. So the incidence rates are represented in the observed incidence rates are represented by the dots, and then the estimated curve is represented by the blue curve. Okay. And so you see that the curve on the left is greater than or equal to the curve on the right for all doses. So next slide. Okay, so this slide gives you the estimates of the level 1 BMD05 and level 2 BMD01 with simultaneous 90% confidence intervals. So the estimates there to the level 1 BMD05 was 215 mg per kg per day, and level 2 BMD01 was 234. So the level 2 BMD01 was slightly higher than the level 1 BMD05. And the far right column provides the 90% confidence intervals. So they're expressed in terms of, so the first number there for each BMD is the lower limit, and then the second number is the upper limit. And so the lower limits that are bolded, those are what we at EPA call the BMDLs, and they are used as the point of departure. Now the important thing to note here is that the confidence intervals provide 90% simultaneous 90% coverage. So that means that probably that both intervals simultaneously contain their respective parameters is 90%. Okay, so that's important because basically what that says is, so if we had done each of those confidence intervals individually and separately and not jointly, what that would mean is that the probability that each one contains its respective parameter is 90%. But the probability that they both contain their prospective parameters at the same time, which means the intersection of that event could be less than 90% and probably would be. And so and so we could basically, it would be difficult to choose which one of these two BMDs to use for the critical effect in that case, because you'd have less coverage if you choose after you've done the analysis. But because we computed them simultaneously, we can make the decision on which BMD to use as the critical effect post hoc, which means we can run the analysis and look at the results and then decide which one of those BMDs will have as the critical effect. And we won't lose coverage. We'll have the same coverage. So I can choose the lower one if I want, like the 111, the first one, and I still maintain the 90% coverage. Next slide. So basically, this is just the advantage of this ordinal analysis. You can estimate the two BMDs based on different salivary levels in one analysis. So you only have to model the data once. And it's also flexible. You can we don't have to have just two levels. We can have more than two levels if we want. We just did two levels for this analysis, but you can use more. And that's all I had. Thanks.
Excellent, thank you, Todd, and thank you very much to our speakers. What we'll do now is we'll go to the written questions, and I'll put these forward to the panel, and we'll have about 10, 12 minutes, and then we'll wrap up. So, I think the first question, I'll direct this to Beth and Erin. So thank you for your presentations. As I understand it, your systematic reviews to some extent were a test run of the Irs assessment methodology as it is evolved to now, right? So obviously that's been under revision in the last few years. The draft of the Irs handbook is now publicly available and is under review. Can you comment on how the Irs approach has changed since your systematic reviews, and then how the experience you had in conducting your reviews may have informed those developments? So how this might look in the future as well.
Okay, I'll take this. The big changes that I can think of off the top of my head are in terms of software that we use to conduct the reviews. So when we did the phthalate systematic reviews, we did all of our title abstract screening just using Excel spreadsheets that we shared back and forth, and it was awful. And things have gotten a lot more efficient since then, where we now use distiller software typically for screening. We also have adapted, in some cases, Swift software. If we have a huge database, we can use that to narrow it down to just the health outcome studies that are most likely to be relevant to the Irs assessment. And then using, we have Tableau for our evidence maps, and then we have the Hawk software that we use for the study evaluations and for extracting data and making figures for the assessment. So that's huge. That makes things so much easier on us. And one big thing that some in Irs are working on is making the data extractions into Hawk more consistent in terms of the terminology that's being used, so that I'm going to explain this terribly, but just like just to help with consistency, it helps with our figures, and it was definitely an issue with the phthalates assessments. We really struggled with how to extract data into Hawk in a good way, and that's gotten a lot better since then.
Yeah, we're calling it the Controlled Vocabulary Project, where people are develop predefined terms that are applied, and it makes the data extraction a lot cleaner and a lot more consistent. And it sounds like a really little thing, but it really makes a big difference, especially with how Hawk pulls data for making visualizations. And then one of the other things specifically going on with Hawk related to study evaluations is when the study evaluations were done for the phthalates project, there was a limitation in Hawk where you could only put a single judgment for the domain. And so they didn't necessarily always reflect the outcome level judgments. But pretty recently, I think it's in the last couple of months, they've made a big update to HAWK where we can now put outcome level, different judgments for the outcome level differences in HAWK, which has been great.
Yeah, and I'll just say that, it's really just been a process of small improvements. I think every time we do a systematic review, we learn something and we then go back in and we tweak, we realize some bit of language wasn't clear to people, and so we go and we tweak it. And so if you look at our process now versus the phthalates, it's fundamentally the same, but we have made lots of little improvements that have, I think, made a big difference in how difficult it is to do the systematic review, and I think transparency for the reader.
Yeah, so we actually had quite a few questions from participants in the Q&A about the methods involved in making systematic review more efficient and the types of software tools that you use. So maybe, because we threw around a lot of acronyms, right, for like HAWK and Distiller and Sciomes, Active Screener and things. Maybe, can you go from like top to bottom maybe, and just quickly introduce maybe the major tools that people might want to look up if they're conducting their own systematic reviews in the near future? So from, say, screening through HAWK as a data repository, maybe Distiller as a way of handling data throughout a systematic review process.
Yeah, so we start with our database searches. You have all of your studies, which as I mentioned, it could be like 10,000, tens of thousands of studies. So if you have a huge database like that, the first thing you would do, we use Swift software. Now I'm blanking, is it Swift Active? Anyway.
Swift Review, I think, can be used for sort of applying tags and, binning studies. And then Swift Active Screener is kind of that machine learning, it can, you can get to that threshold where the machine can take over.
Yeah, okay, yeah, that's it.
But only for literature screening. I want to be very clear about that we do not use machines to do our systematic reviews.
We have not, no. So that's screening step. Like I'm working on the vanadium assessment. We had like 25,000 studies, and by using that binning, we got down to like 3,000 studies that were actually relevant to health effects. So that's huge, having that. So Swift, then that would go into Distiller. Distiller SR is a screening software where you can have two independent screeners working simultaneously to get through your screening efficiently. And you can also use Distiller to extract data for an evidence map. So we use that. At that stage, the evidence mapping goes from Distiller into Tableau to visualize it and make it public. And then from there, we go into HAWK. So that's an acronym for Health Assessment Workspace Collaboration, I believe. And that was originally used at NTP, I believe. And we hired the guy that developed it, Andy Shapiro, who's on our staff now, and he's tweaking it all the time to make it better for Iris. So that's our study evaluations and data extraction.
Yeah, so HAWK's actually also very interesting because it's the way you make the individual decisions throughout the systematic review process, where you've got the data that you've extracted, and then the analysis that you're doing, a risk of bias assessment. All of this data is in HAWK and is accessible to any user, if I understand correctly. Once the publication has been approved, then people can link back to that and see everything that you did in that evaluation process.
Yep.
Yeah. And then that data, of course, is also theoretically reusable by other researchers. So if you have similar data extraction requirements, it may not be necessary to repeat the same data extraction that you will have already done, or at least you may only need to do some additional supplemental data extraction and not have to start from the beginning again.
That's correct.
Excellent. Good. So we also had a question. I can find them again. So let's see. So someone, Catherine Kelly, asked how common it is to exclude papers that aren't written in English in systematic reviews, and whether or not there's a risk that important information might be in those papers but may go missing from the review process. So, I mean, as an editor of systematic reviews, this is something that we do see sometimes, like particularly with an increasing amount of research from Chinese universities, for example, that may be published in Chinese but not English. Do you have any way of handling that, or is it something that's for future work? Or
we make an effort to obtain and not necessarily translate the whole paper, but translate the title and abstract and see if it's PICO relevant, and if it seems as though it is and would make an impact on the assessment, then we will go ahead and get the whole paper translated.
And I think we've had instances where we've found non-English papers where they'd also published the same data in an English journal. And so during the initial stages, we were able to find, oh, actually, we didn't need to translate this because we have this other paper that has that same data. It's that same study. So I think that's happened as well too. But yeah, we try and get all data that's out there.
good. And then, so we also have a question here about the process of contacting primary study authors for data when it's not been reported in a study. So you have, say, data on request from the authors. What's your experience been of trying to retrieve kind of, when you're doing a systematic review, this sort of information in terms of, say, the amount of time it takes to do, and then maybe how successful you are in doing it? Because again, in other systematic reviews, like the World Health Organization systematic reviews on burden of disease that we're handling at the moment at the journal, they've found it extremely difficult after the event to get usable data from study authors.
My experience is about 50-50, I think, response rate. We have kind of a pre-formatted letter that we send out. We give people 4 weeks to respond, and we contact the corresponding author on the paper. And we only do it when we think it's going to make a difference with our study evaluation ratings. We don't do it for every single deficiency in a study. So we're kind of, I guess, we have to be pragmatic about how we prioritize that. And I know for some of the larger assessments, like things like PCBs, I feel like they're setting a date limit on contacting authors, like they're trying to contact people from the 50s. You just got to draw a line somewhere.
We're much more likely to hear back if it's a recent paper.
Yes.
And sometimes we even will ask for additional analyses, and we're very appreciative when authors can do that for us. And sometimes we ask a little question and never hear back. So please help us if we could email you.
Right. Well, thanks very much. I'm afraid we are now, unfortunately, out of time. So I'd like to thank our presenters for their hard work, the excellent systematic reviews, and taking the time to present this to us today. Obviously, there are a lot more questions that we could cover in the discussion. So anyone with questions should follow up with EBTC, and we can put you in contact with the systematic review authors afterwards. There are links which we will send to all the participants to this video being posted on YouTube. Useful links that have been shared through the chat, so you can follow up on those. And also information about the next online symposium we'll be organising, which will be on knowledge organisation systems for systematic review, which will touch on issues like the use of platforms such as Hawk. We again want to have to apologise again for the technical difficulties we had the audio, and we'd like to thank you for your time and your patience and your interest. So goodbye.